Very good. Today's lecture is basically to clarify the cell cycle regulations and the molecular pathways involving in that regulations. This is a very important to know the basic checkpoints and the regulations, regulatory proteins which involves in the regulation of cell cycle progression. Basically, we know cell cycle, cell division of two types. One is mitosis, one is meiosis. Today's lecture is basically the mitosis process. The basic mitosis process you all know, and the steps. Firstly, you should know the steps of mitosis. Remember all those steps. One is G0 phase, which is called gap phase. Gap phase. It is variable. And it depends upon the actively division of a cell. This means how the cell reacts during the cell cycle. Then comes G1 phase, then comes S phase, then comes G2 phase, then comes mitotic phase. In case of G0 phase, I want to say again that this phase is variable for different cells. How cells actively differentiated, it depends upon this G0 phase. Then comes the G1 phase. G1 phase, approximately we assume in a mammalian cell, this mitotic complete process takes 12 to 24 hours. Mean should be 16 hours. We assume that. Within that, 5 hours, the cell within this G1 phase, within this 16 hours complete cell division. 5 hours takes in G1 phase. Then comes the S phase, the synthesis phase, where DNA synthesis occur. In G1 phase, there is RNA, protein, and organelle synthesis seen. In S phase, there is DNA synthesis. This lasts about 7 hours. And G2 phase, it lasts about 3 hours. And mitotic phase, 1 hour. But this is not my prime points. My prime points is G1 to GS phase, there is a checkpoint. And G2 to mitotic phase, there is a checkpoint. these are my prime important area. This video is definitely about cell cycle, but I want to target these 2 points which are very important for a cell cycle. Now again, I want to recap these things completely from a point G1 phase, that is a gap phase, which defines whether the cell is actively dividing or not, and it is variable. G1 phase lasts about 5 hours, within that 16 hours complete cell cycle. Here RNA, protein and organelle are synthesized. In S phase, DNA synthesis occurs. It lasts about 7 hours within 16 hours. And G2 phase, it lasts about 3 hours. And the mitotic phase, which lasts about 1 hour. There are 2 checkpoints between G1 and G2 to mitotic phase. Here, these checkpoints is regulated by cyclin and cyclin dependent kinase protein. Cyclin dependent kinase complex. Here is S cyclins. I will come in details with these cyclins. And here is also a checkpoint which is also M cyclin dependent regulation. Now I will come in details with these cyclins. Comes to the checkpoints and cyclin dependent kinases. When cyclin with cyclin dependent kinases attached, this is a complex, then only this thing activates. This called active. It promotes mitosis. In G1 to S phase, here cyclins are D and E with CDKs, cyclin dependent kinases. Here the cyclin dependent kinase are 2. This is an isoform where S to G2 to M phase, here CDK is 1 with cyclin A or B. This is a promoting factor. These are the promoting factors for mitosis. There are some inhibitory factors which regulates the cell cycle. One is called cyclin inhibitory protein kinase. And there are 2 important proteins which are called APC-C, which means anaphase promoting complex oblique, this C defines cyclosome, and there are other protein also which is act like this. Here is a protein complex. Here is the F box position. Here is the RBX1. Here is the CUL1. Here is E2 component. Here lies the ubiquitin component. Ubiquitin component. Here comes the phosphorylated part of that ligand. And it will get arrested with this F box. These are all ligases which causes ubiquitination and arrest the mitotic process. It's arrest the mitotic process. This ubiquitination arrest this complete process. These are the checkpoints proteins. These are all ligases which causes ubiquitination with the ubiquitin moieties with that F box culin and RBX1 E2. These subunits completely arrest the protein and destroy it. And there is anaphase promoting complex with cyclosomes which also acts like these. These are all ligases. And there is cyclin inhibitory protein kinases which also inhibit this kind of cyclin, the S cyclins or same cyclins. These are the regulatory part of the cyclin dependent kinase with cyclin attached complexes. Without cyclin, cyclin dependent kinase is inactivated. Without cyclin dependent kinases can't work. Always remember that. Now I will come. Now we comes to that main part, that checkpoint regulation part. Firstly, clarify the things when cell size and cell count. One is the thing that increment of cell size and there is division. When there is division, there should be cell divide process and the divide process and cell survival. But increasing in size, there is proliferation cell growth, division, cell survival. In case of any organ or tissue, when it proliferates, it has to be grown, it has to be division of cells, and the cell survival rate of those cells which divides. Three basic main factors depends upon this kind of organ development. Now comes this cell division depends upon three things. One is mitogen, which is the which influence the cell divisions. Secondly, growth factor. Third is survival factors. Mitogens are basically, we can say, platelet derived growth factor. There are different growth factors. Helps or promotes mitosis. How mitogen promotes mitosis, these growth factors, I will come in details. But the overall growth factors are insulin like growth factors 1, 2, neural growth factors, there are several. And in survival factors, to arrest the apoptotic cell death, the programmed cell death, there are inhibition of BCL2 gene, or there is TNF alpha. These are important factors for survival factor in case of mitosis, programmed cell death. Now comes to the growth factor induced mitogenic activity in cell division. The mitogenic activity means the platelet derived growth factor, any kind of growth factor when comes to a cell membrane. Assume this is a cell membrane. There are two subunits. Here comes the growth factor. When it attached to these subunits, growth factor receptor, this is a growth factor. The tyrosine molecules within this growth factor moiety get phosphorylated. There are some breach proteins which are then activated by this phosphorylation of tyrosine molecules of growth factors. These are bridging proteins. Which in turn phosphorylate the inactive RAS protein to active RAS protein. Here there is inositol part, PI2. Here there is 3-phosphate with inositol group, PI3. It comes to this by GTP to GDP by hydrolysis. Here this RAS protein is anchored to the cell membrane by farnesyl protein anchor. This activated RAS, active RAS protein comes to different two pathways. One is PI3, AKT, mTOR. Here RAF, these two pathways activates a gene which is called MYC. It's a transcriptor regulatory protein complex. From the beginning, starts from the beginning. When the growth factor attached to the growth factor receptor within the mammalian cell membrane, there is tyrosine molecules within that growth factor receptor which are phosphorylated. With phosphorylation with those things, there are breach protein which activates the inactive RAS proteins, which RAS proteins are attached or anchored with these membranes with farnesyl moieties, farnesyl protein moieties, and it activates to RAS protein. There is GDP which in turn hydrolysis and comes back to this PI3, PI2. There is dephosphorylation, and then this active RAS protein comes in two pathways, phosphatidyl inositol 3 pathways, AKT protein. AKT protein means which comes, the AKT, you should know, this is called AK strain transforming protein. AKT means AK strain transforming protein. Then comes the mTOR part. mTOR means mammalian target of rapamycin. These are the MYC. MYC is a transcription regulatory protein. Which is also called master regulator of cell cycle entry and proliferative metabolism. PI3 phosphorylated inositol 3 kinase. All are kinase activity, mainly phosphorylation of all proteins. RAF means rapidly accelerated fibrosarcoma protein. This is called mitogen activated protein kinase. The RAS protein, you should know what is RAS protein. This is a small GTPase. These are nothing but the small GTPase. These are all small GTPase protein. RAS comes from rat sarcoma virus protein. You should know the all names where it comes from, because those are important. In books there is always written RAF, RAS, PI3, AKT, mTOR, MYC. You should know the name of these proteins. Where it comes from, it defines those abbreviation. AKT means AK strain transforming protein. mTOR is a mammalian target of rapamycin protein. RAF is a rapidly accelerated fibrosarcoma protein. MAPK is a mitogen activated protein kinase. MYC is a master regulator of cell cycle entry and proliferation metabolism protein. Here PI3 phosphatidylinositol 3 kinase. And here you should remember this RAS protein is anchored within the cell with the farnesyl protein anchor moieties. This is the main thing. This continues cell cycle by activating retinoblastoma, suppression protein. Which in turn activates E2F gene expression and continue the cell cycle. If this is blocked and sustained MYC expression, patient may undergoes carcinomatous changes. From the beginning, I should say, when the growth factor attached to the growth factor receptor within the outside the cell membrane, inside the cell membrane, the moiety, the parts of growth factor receptor, there are tyrosine molecules within them. Those are get phosphorylated. With the bridging protein, it activates the inactivated RAS protein, which is anchored with farnesyl proteins with the cell membrane. It activates with the active RAS protein, where it activates some molecular pathways within the cell. Those are PI3, AKT, mTOR within the RAF, MAP kinase 2, ultimately expressed those proteins. Those are called MYC proteins. Those are the master regulator of cell cycle entry and proliferation metabolism, which in turn activate the retinoblastoma suppression proteins, which expresses the genes E2F. These expressions continue cell cycles. Where these are blocked, the carcinomatous changes happened. the sustained activation of MIT expressions. This is the basic pathway, but you should remember another important factor point. You should remember when cell get injured, then there are two proteins expressed. One is called ATM, one is called ATR. What is ATM? ATM and ATR signal to the cell that cell should stop their divisions during the cell injury or DNA injury. ATM, the full form is ataxia telangiectasias mutation gene, and other is ataxia telangiectasia RAD3. These are two important molecules, signal molecules, which signal to the cell to stop cell division during injury of the DNA or cells. These are the things which I want to clarify today. Thank you.